We are always privileged to have you at Medical Sciences, and in this session we want to discuss about antimicrobial resistance. Antimicrobial resistance comes in whereby the drugs which were initially resistant to a microbial agent, or what was maybe it was killing 99.9% of the bacterial or the antimicrobial organisms, it is no longer function. That is what we are calling resistance. So once the microorganisms which were susceptible or which were sensitive to this drug, at a later or at the long run, they become inactive or they do not respond to this medication. And there are different types. As we are beginning, we always remind you to subscribe, share the video, and like. So there are different types of antimicrobial resistance. Types of antimicrobial resistance, they include, so here we see how are they acquired. So the types of antimicrobial resistance, we have what we call natural resistance, we have what we call acquired, we have what we call cross resistance, and we have what we call multi-drug resistance. And under natural, here we see there are some bacteria or there are some microorganisms that they are naturally resistant to certain drugs due to their target site. For example, we see gram-negative bacteria. They are naturally resistant to vancomycin because vancomycin targets only the cell wall, the peptidoglycan layer of the cell wall of the gram-positive. So we see gram-positive like staph and enterococci being active to vancomycin, but gram-negative bacteria, they are naturally resistant to vancomycin by their nature or by their structure. So structurally they are resistant. Then others which are under acquired, we shall see how resistance is acquired either by mutation or by transformation, whereby the bacteria or the microorganism acquires the naked DNA from the environment. That is transformation. Then we shall see transduction, whereby this one is via the bacteriophage, whereby the bacteriophage is the one that transmits this resistant gene to different. And lastly, we can talk about conjugation. This conjugation is whereby two bacteria, they share their genes via sex pili. They are sex pillars. So this is acquired. So we have four types. Then under cross resistance, this happens in drugs with the same chemical structure. When drugs have the same chemical, when the drugs have the same chemical structure, we see them being resistant. For example, the penicillins and the cephalosporins all have they target the same, that is penicillin binding proteins. And because they target the same, so there is cross resistance. That an organism which is resistant to penicillin can be the same resistant to cephalosporins due to cross resistance. Then multi-drug resistance, this is where we see multiple drugs or different drugs. Here, different drugs having the same mechanism. The way we are seeing it in the video on mechanism of action. So if there are different drugs, different chemical nature, but having different mechanism of action, we see them being resisted. That is what we call multi-drug resistance. So this is the different types of drug or antimicrobial resistance. whereby we have natural acquired, then cross-resistance and multi-drug resistance. And after seeing how it comes into existence, we want to look at the mechanism of resistance. We want to look at the mechanism. We want to see how do different microorganisms become insensitive or become resistant to certain drugs. And one of the mechanisms we are going to see alteration of target site, We shall talk about decrease in permeability of the cell membrane or cell wall, decrease in permeability. We shall see production of inactivating enzymes. Then we shall look at the development of efflux pumps. Then lastly, we shall look at alteration or production or development of alternative pathway. Development of alternative pathway. What we call development of the bypass. So these are the major 5 mechanisms of drug resistance, whereby number 1, which is alteration in the target site. Alteration on the target site, we see different microorganisms, especially like we have fluoroquinolones, and these fluoroquinolones, they do target DNA gyrase, which we call topoisomerase 2. So any alteration, any mutation, if mutation occurs, if there is mutation in the gene responsible for the synthesis of DNA gyrase, then this microorganism will become resistant to fluoroquinolones like cipro and levofloxacin. So this is what we are calling alteration of the target site. Target site, if a drug was targeting some site and there is mutation or change in genetic makeup, it will no longer respond. And an example is the fluoroquinolones that target DNA gyrase. So any mutation that occurs on the DNA gyrase, it can no longer recognize it and it becomes resistant. Then number 2 is what we are calling decrease in the permeability of the cell membrane or the cell wall, cell membrane stroke cell wall, whereby in decrease in permeability, especially the gram-positive bacteria, whereby they decrease in the size of pollins or pores in the cell wall. And when they decrease the size of the pores in the cell wall, drugs which would enter via the cell wall, they cannot enter. So if this is the cell wall, this is the cell wall. So this one, if this was the size of the pore, this one acts by decreasing. It decreases the pore size so that the drug which was entering can no longer enter. So that is decrease in the size of the pore, which brings about decrease in permeability. And this one results into, because the drugs are not entering, you cannot achieve the therapeutic dose. The therapeutic dose is not reached because of decreased entry of this drug. That is what we call decrease in permeability. Then the third one is production of inactivating enzymes. And these enzymes, we have seen it peculiar of production of beta-lactamase, which we call penicillinase. Penicillinase enzyme, This beta-lactamase is the one that destroys the beta-lactam ring. These ones destroy the beta-lactam ring of penicillins, beta-lactam ring of penicillins. Drugs like penicillin, drugs like cephalosporin, drugs like carbapenems. So due to production of this enzyme penicillinase, which destroys the beta-lactam ring of these drugs, they render them non-functioning. And we see even there is production of another enzyme hydrolase, or what we are calling elastase in macrolides. So majorly we see this inactivating enzymes making these drugs non-functioning, hence they can survive in their presence. Then number 4 we can talk about in this session is the development of efflux pumps, pumps like ABC transporter, pumps like MDR pump. These ones, whenever the drug enters in the cell, before it accumulates, it is pumped out. So the bacteria have developed a mechanism that whenever the drug enters inside the cell, immediately before it accumulates, it is pumped out of the cell. And that is what we are called. So this one also prevents the accumulation of the drug to reach the therapeutic dose. So decrease in permeability and development of efflux pump, they cause resistance by decreasing the concentration of the drug inside the target organ. So we cannot get the therapeutic dose or the pseudo dose or the lethal dose to kill the bacteria. So we do not get the lethal dose. This one we can call it lethal dose to kill the bacteria. So that is what development of efflux pump does. Then lastly, which is number 5, is development of the alternative or development of the bypass. This one is a peculiar of drugs like sulfonamides, whereby we saw that in presence of PABA, we form dihydrofolate, and this dihydrofolate is converted to tetrahydrofolate. And we saw here there was a synthetase enzyme, and this one was a reductase. This one were inhibited by different drugs like sulfonamides and trimethoprim. So what happens is, if trimethoprim targets this synthetase enzyme, so the bacteria develops a mechanism to bypass this process and converts PABA directly to tetrahydrofolate. This is what you're calling bypass mechanism, a peculiar seen in sulfonamides. Sulfonamides and trimethoprim, whereby the bacteria develop a bypass. In case a drug was targeting this step, the bacteria develops a mechanism of skipping it, and it continues to survive in the presence of that drug, the peculiar of sulfonamides and trimethoprim. So thank you so much for listening from the beginning until the end, where we have discussed about mechanisms of drug resistance, whereby we have said there are majorly 5, that is alteration of the target site, decrease in permeability, production of inactivating enzymes, development of efflux pump, and development of alternative pathway. Thank you so much. Always remember to subscribe if you haven't subscribed, and share your comments. Thank you so much.